Okay, this is a special tutorial in Chapter 4 on the Bombay phenotype. And the reason I'm doing this is because in the audio lecture I didn't have screen capture, so I really couldn't fully explain this Bombay phenotype to you, but now I have my cursor and I can draw on the screen, so I'm hoping this little tutorial will help you. If you recall, the Bombay phenotype is our example of what's called epistasis. Epistasis means when you have two or more genes, specifically in this case two genes, two separate genes, which are both impacting the same trait, and where one gene can actually block expression of the other gene. So phenotypically you won't see expression of a gene because of the second gene is blocking it. So that's what epistasis is. And this is an example from that. Bombay phenotype was first described by a person from Bombay, so that's where it got its name. You don't have to be from Bombay to have this. It's rare, but it does exist. So what we have here would be the phenotypes of these individuals, blood types. And the way blood typing is done is by really using antibodies to look for antigens on the surfaces of cells. Antigens are glycoproteins. And glycoproteins are made by making a protein on the ribosome, and then in the rough ER lumen there's an enzyme that attaches a short carbohydrate little string of sugars onto that. So if you have type A blood, then you have an enzyme that adds the type A carbohydrate to the protein. If you have type B blood, you have an enzyme that makes a B type sugar chain that puts it on the protein. But the thing is that the enzyme which makes the sugar chain, that's the A or the B or the O. But if you have, but there's another gene that's needed here, and you may not have ever heard of this, but the gene is called H, the H substance. The H substance is the protein. So I'm going to draw a red blood cell, could be any cell. There's a transmembrane protein, the H protein, and then attached to that will be the little sugar chain here. So pretend like that's the A antigen. It has a protein and a sugar chain. The B antigen might have the same H protein but then a little different sugar chain. So I'm just making up the sugar chains, but they're supposed to look different. So if you have type A blood, your cells only make the A type glycoprotein. If you have type B blood, your cells only make the B type. If you have A and B, your cells can make both, and both of them can be spread out on the cell. If you have type O blood, usually that means your cells make the H substance but no sugar chain is attached. And so even though you'll have typically H substance on your cell, there's no sugar chain, and so that's usually nothing is really detected by the immune system. But in any case, so what we have here would be, for example, these individuals, somebody type A mates with somebody type AB, and it would seem that this person is completely impossible. How can you have a type O? But remember, type O is just the phenotype. So it turns out that these individuals, let's say this individual would have one A allele and one O allele, and this one has one A and one B. Then when you mate it, you might get somebody who has this individual in their nucleus, in their cell nucleus, may have one A and one B. But the other thing that's also happening in this cross at the top is that you have probably what's called a big H, little h, a heterozygous genotype for the H substance. Remember, the H substance is the protein component. The AB is the enzyme which adds on the sugar component. So if the genotype of these two parents was big H little h crossed to big H little h, isn't it possible to get this child who could have the alleles for A and B, but then also have two little h alleles? Now if you get two little h alleles, that means the cell is not making any of this protein. And if you don't make the protein, then it doesn't matter what enzymes you have, there's no protein to attach the carbohydrate onto, and therefore nothing gets displayed on the surface of the cell. So in the nucleus you may have the alleles for big A or for A and B, but because your body can't make the H substance protein, then that can never be displayed. However, the A and B allele can be passed on to the offspring. And assuming this partner has, usually normal would be big H, then when you do this cross, these individuals down here would be heterozygous, so they would show their blood antigens because they would be able to make the H substance. So this is really the genotype of this individual, but O is the phenotype. And so when you have somebody like this, that's an unusual case where the H genotype is blocking expression of the AB genotype. So occasionally you'll see, something posted on the internet where somebody says, I have type O blood, one of my parents has type AB. How is this possible? Well, there's two answers to that. First, maybe that parent is not really your parent, or in a rare occasion it could be this. So at least there's some hope that that person has, a legitimate genetic reason for looking O when it appears that they couldn't possibly get that from their parents. So anyway, a little tutorial. I hope this helps, and let me know if you think this was helpful.